Hello, my name is Jacob Blue, and today I'm going to be talking about my investigation of voltage-gated proton channel interactions with cytoskeletal motor proteins in a model of male infertility. Spermatozoa or sperm are male reproductive cells, which propel themselves by whipping their flagella. Symmetrically localized calcium ion channels known as catspur activate flagellar motors for movement. Catspur are activated by an alkaline gradient produced by asymmetrically positioned proton channels known as HV1, which extrude protons from the cells. Considering the dynamic fluid mosaic model of plasma membranes, the mechanism by which HV1 is asymmetrically anchored is unknown. Evaluation of human testes protein libraries demonstrated a potential interaction of HV1 with peptide fragments of dynein light chain TEC-step type 1 protein, abbreviated as DynLC1, which is a cellular cargo adapter for motor proteins. Recent fertility screenings have shown absence of DynLC1 from infertile sperm flagella and relative abundance in fertile sperm. The aim of the study was to determine whether native state DynLC1 and HV1 proteins physically interact with each other. This is determined using co-immunoprecipitation of hemagglutinin HA-tagged HV1 and MEEK-tagged DynLC1 directly pulled down by anti-HA or anti-MEEK antibody respectively. If HV1 and DynLC1 interact with their native states, then pull down with either antibody should bring along the other protein together as a pair. Experimental co-immunoprecipitation was performed in 2 iterations of anti-HA targeting HA-HV1 or anti-MEEK targeting MEEK-DynLC1. Co-immunoprecipitation studies were visualized by Western blot. Staining with a primary antibody opposite of the pull-down antibody allowed visualization of interacting proteins. Staining with the same primary antibody as the pull-down antibody ensured positive controls. Non-immunoprecipitated cell lysates served as negative controls. Western blot analysis indicated that anti-MEEK pull-down brought along HA-HV1, visualized with anti-HA stain. Western blot analyses also indicated that anti-HA pull-down brought along MEEK-DynLC1, visualized with anti-MEEK stain. The Western blot analysis indicated that HV1 and DynLC1 associate and interact with each other. Pull-down with either anti-HA or anti-MEEK produced MEEK-DynLC1 and HA-HV1, along with the originally targeted protein. Some limitations of the study include relatively faint Western blot experimental bands and unexpected experimental bands at molecular weights greater than 35 kilodaltons. This has been analyzed in further detail. In conclusion, HV1 and DynLC1 have been shown to interact in their native protein states. However, additional experimental protocol optimization is required to ensure unequivocally variable results. Further research entails studies with the Z sperm to evaluate mutations in HV1 and DynLC1 related to male infertility. Applications of this research include development of new male fertility treatments and pharmacological contraceptives disrupting sperm-specific HV1 activity. Thank you for your time and consideration.